At the cellular respiration change machine, inputs of molecules such as amino acids, fatty acids, or glucose are broken down into energy. For our uses, the $1 bill represents the amino acids, the $5 bill represents fatty acids, and the $10 bill represents glucose. The $10 bill receives the most change back because glucose is broken down into the greatest amount of ATP. The machine acts as the mitochondria. However, not all the processes take place in this organelle. Glycolysis happens in the cytosol, and the end byproduct, pyruvate, is transported into the mitochondrial matrix. Unfortunately, our change machine is a scumbag, and like the actual process, requires an input of 2 ATP molecules, which we will depict as pennies. Glycolysis begins with a molecule of glucose, and through a series of 10 chemical reactions, is broken down into 2 3-carbon molecules of pyruvate, 2 net ATP, and 2 molecules of NADH. ATP during glycolysis is produced by substrate-level phosphorylation, which is the addition of a phosphate to ADP. During acetyl-CoA synthesis, the pyruvate is oxidized and splits off to form carbon dioxide. During this, electrons are donated to NAD+, to be reduced to form NADH. The remaining part of the pyruvate group is transferred to coenzyme A to form acetyl-CoA. Therefore, during acetyl-CoA synthesis, 2 molecules of CO2, 2 molecules of NADH, and 2 molecules of acetyl-CoA are produced. During the citric acid cycle, the remaining carbon molecule is completely oxidized. The citric acid cycle is a series of 8 reactions, and at the end, the starting molecule, oxaloacetate, is regenerated. During reactions 3 and 4, 4 molecules of CO2 are released, for a total of 6 during the entire cellular respiration process. In addition, throughout the reactions, 6 NAD+ molecules are reduced to form NADH, along with 2 FAD molecules that are reduced to FADH2. During the electron transport chain, electrons in the electron carriers are released during redox reactions through complexes 1 through 4 in the inner mitochondrial membrane. This causes the transfer of protons as well as creating a concentration and charge gradient. Electrons donated to NADH enter in through complex 1, and those donated by FADH2 enter through complex 2. As electrons pass from donors to acceptors, the energy of the electrons is reduced. The transfer of electrons causes an accumulation of protons in the inner membrane space. Since protons cannot passively diffuse across the membrane, ATP synthase helps. Protons flow through the F subunit 0 channel, causing it to rotate, allowing the F1 subunit to catalyze the formation of ATP from ADP and phosphate. During this process, 10 NADH molecules and 2 molecules of FADH2 produce 28 molecules of ATP.